Today I'm going to be talking about integration of RNA-seq and chromatin accessibility to look at genome-wide expression. So this primer has been really nice. It's given us a really great overview on RNA-seq, and today we're going to take it kind of an extra step by talking about chromatin accessibility. So that's going to be the majority of my talk today, and then how we can integrate this kind of data into RNA-seq analysis. And so as this is a primer, I'm going to give everyone some background information about chromatin in general. So DNA packaging into the nucleus is really crucial for regulation of gene expression. And I like to think about this as when I have to take down my Christmas lights every January, and if I wrap them around kind of in my elbow and put them away in a nice organized manner, it's much easier for me to take it out next Christmas, right? But if I just kind of shove the lights in there and don't really think about how I'm storing them, it's really, these lights are really easily breakable, and it's really hard to use them later next year, right? So I kind of like to think about packaging of DNA into a nucleus in the same way, because we have 6 feet of DNA that we're trying to pack into a tiny nucleus of a cell. And so we have our double helix that eventually needs to be wrapped around histone proteins, and this is called a nucleosome. These nucleosomes then need to be further condensed into chromatin fibers and then further condensed into chromosomes, right, which are kind of the backbone here of our nuclei, right? And so we have our closed chromatin here. So DNA that is wrapped around a nucleosome is referred to as closed chromatin. And chromatin in this state is generally silenced and is not being actively transcribed, right? Because the physical space of Pol II, you can't get into the DNA here and you can't activate transcription. However, chromatin that is open, and you have your nucleosomes in a much more relaxed state, and DNA in here is amenable to modification and activation by Pol II or other histone modifications, transcription factor binding. And so regions that are open are more likely to be active. And so I'm going to talk about closed chromatin and open chromatin today in the context of Atec-seq. So we have a lot of different ways to look at the chromatin landscape. We can look at open and closed chromatin by Atec-seq. We can look at where proteins are bound to DNA via Chipseek, and then we can look at long-range chromatin interactions via Hi-C. And I'm going to touch briefly about each of these techniques, the pros and cons, and how we can integrate these with RNA-seq to determine gene expression patterns. So Atec-seq is a relatively new technology. It arose in the mid-2010s, and it is a way to look at regions of chromatin that are open in a non-biased way. And so what we do is generate nuclei from a small number of cells. Only about 50,000 cells are required for this technique, which is very few, I would say, and it's actually a really big benefit to this technique is that you don't need a lot of cells to do this. So you take about 50,000 cells, make nuclei from these cells, and then from there we can isolate our chromatin. And when we have our chromatin, then we add this TN5 transposase, which has adapters that are attached. And this is a transposase method, so this transposase will integrate into regions of the genome that are open, right? Because it physically can, right? So these regions are, the transposase is integrated in, and then this will fragment our DNA into small pieces, shown here. So these pieces have the adapters attached from the transposase integration, and we can isolate these small fragments of DNA with adapters and perform sequencing on these. This process is pretty short. It takes only 3 to 4 hours plus the time for sequencing and analysis. And importantly, it can be performed on either fresh or frozen cells, which is again a pretty big benefit to this technique. The resolution of our DNA that we get here is on the order of nucleosomes. And so when you look at your DNA library on a gel or a tape, you want to see this nice striated banding pattern of fragments. And so this is a good quality control check for Atec-seq library preparation. You want to see this nice striated banding pattern to show you that their transposase is integrated at a nucleosome level resolution. So I'm going to switch gears just a little bit and talk about how we prep our chromatin for Chipseek, and then I'm going to talk about processing the data a little bit later in the talk. So Chipseek is a little bit different than Atec-seq in that here you cross-link your cells before you generate nuclei and isolate your chromatin. So the purpose of cross-linking is that you want to cross-link your protein of interest to the DNA in which it's bound, right? Because you're going to then isolate this protein and the DNA that's bound with it. So first thing you want to do is cross-link your cells, and you want to make sure that you do this experimentally. So you want to make sure that you don't overfix your cells, because that leads to chromatin that's too big and therefore unusable for sequencing. So once you cross-link your cells, you then sonicate your chromatin. So it's a mechanical method of shearing, which is different from the Atec, which is transposase mediated. And again, you want to make sure to kind of troubleshoot this by determining the length of time and the amount of cells that it will take to get your DNA to about 200 to 700 base pairs in length. So here's kind of an example of a time course that I ran when I was first doing these Chipseek experiments, where I sonicated for 4 to 7 to 10 to 13 minutes. And as you can see, the more you sonicate, the lower your DNA smear will be on the gel. However, here would be an example of a not so great shearing, because you have a ton of chromatin here that's really, really big outside of the context of this tape. And so once you play around then with cellular concentration, you can kind of correct for this. And if you shear a fewer number of cells for a lower amount of time, eventually you'll get chromatin that looks like this. So it's a nice little smear of fragments between 200 and 800 base pairs. So kind of troubleshooting this at the beginning will save you a lot of headache later on down the road. So I highly recommend the troubleshooting before you start. So once you have your chromatin to the size in which you would like, the next thing you'll do is immunoprecipitate using an antibody. And this is the other kind of way that your troubleshooting could be really helpful. So you want to use an antibody that is specific to your protein of interest. And this is kind of the main drawback to using Chipseek. Not every protein has a really good Chipseek antibody. So I would recommend looking for ones that have already been published as a first pass. So you can do an IP for a transcription factor shown here, or you can do an IP for a histone mark, such as H3K4 trimethyl, which would be indicative of transcriptional active regions of DNA. And so you can do either a transcription factor chip or a histone chip. And then once you isolate your protein, you reverse the cross-link and degrade the protein, and you're left with the DNA that was bound by your protein of interest. Some other differences between chip and Atec-seq. The chip needs to be done on more cells, so you need about 10 to 30 million cells. But this is heavily dependent on how abundant your protein is in your sample. So if you have a transcription factor, you might need to do a chip with more cells, but if you're doing a histone mark that's widely expressed, then you would need much fewer cells to do your Chipseek. It takes a little bit longer to do this as well. It's about 3 days plus sequencing plus analysis. And you can do this on fresh or frozen cells. However, the cells need to be fixed before you freeze them. So they need to be cross-linked before you freeze. And then the resolution of these fragments is going to be dependent on how small you shear your chromatin. So hopefully 200 to 600 base pairs. So now you have your fragments, right? And you want to sequence them. But first you need to amplify them before you can sequence them. So you can either take your Atec fragments that have your adapters already ready, or your chip fragments and then add adapters via your library prep. You'll then PCR amplify these regions of DNA with index primers and then sequence. Some differences in sequencing that are really important. Atec-seq, you need to sequence at a higher depth, over 50 million reads per sample. And I've yet to find a place where more is not better. So the deeper you sequence, the more peaks that you'll see, because you're looking for regions that are open, right? You're selecting for negative regions that are not occupied by nucleosomes. So the deeper that you sequence, the more of these you'll pick up on. However, for Chipseek, you're enriching for protein-bounded DNA, and so you actually don't need as many reads, only about 20 to 30 million per sample.
And there was a related question.
Sure.
Why does Chipseek need so many more cells than Atec?
Because so Chipseek would require more cells because you're isolating only DNA that's bound by one particular protein, right? There's so many proteins in the cell, and there's so many regions of chromatin that are open, and so don't need as many cells to get a higher, you can do more. You'll just have to adjust your transposase in response to how many cells that you have. However, you just get a minimum, you only need 50,000 cells because you're not selecting for a particular protein of interest. So you can get away with a snapshot of fewer cells.
Great, thank you.
Okay, so the sequencing depth is going to be much greater for Atec-seq versus Chipseek, where you only need 20 to 30 million cells to get an adequate picture of your transcriptional landscape. After you sequence, you then align to the genome. We recommend using BWA-MEM because it's better for gap sequences, but there are other aligners that you can use as well. And then once you've mapped to your genome, there's some other quality control checks that you can do to determine how good how well your experiment worked. So first, you want to look at and remove all PCR duplicates. So if you have too many PCR duplicates, it means that your library was over-amplified and your sample was not complex enough. So you either might want to use fewer amplification cycles or use more cells to get a more complex mixture of DNA. The next, we want to remove mitochondrial contamination. And so we do this for both CHIP and Atec-seq. And this is particularly important for Atec-seq, where earlier versions of this protocol, you had maybe up to 80% mitochondrial contamination. And so if you sequence a 50 million reads and you can only use 20% of them, your library is not complex enough to generate the peaks that you're looking for. So the later versions of this protocol have now brought the mitochondrial contamination from about 80% to less than 10%. So that's great. And so you can kind of get away with the 50 million reads, whereas before you kind of needed 100 to 150 million reads to compensate for that. The next thing you want to do is call peaks, and I have a whole slide on that on the next slide. So we're going to call our peaks. And so this is an example of what this might look like. You have your reads that are mapped to the genome, and you can use different peak callers like max2 or Homer to call to assign this as a peak. And then once you have your peaks, you then can annotate your peaks and assign them to genes. And then you have gene regions that are open for Atec-seq or genes that are bound by your protein of interest if you're looking at Chipseek data, which you can then integrate with your RNA-seq data. So we'll get to that in a few slides. First, I want to talk about calling peaks because this is also another key difference between Atec-seq and Chipseek. So Atec-seq here, there's no input sequence to compare to. So what you're actually doing is using a local filtering system to say, okay, there are enough reads in this region right here compared to the 10 kb or so, or 100 kb, there's a parameter you can set of the sequence around it. So you're saying, okay, this region right here is enriched versus all the regions around it. So we're going to assign this as a peak. So this is done using local filtering, and I use Homer to do this. Peaks can be broad or narrow. They can be broad if you have a whole region, a whole gene that's open and actively being transcribed. These promoter sequences are usually, or the regions around the promoter are generally very open, so you're going to get a lot of reads around there. They can also be narrow because you have maybe a transcription factor bound at an enhancer element. And so that's going to be a much smaller stretch of DNA. So you just have to keep that in mind when you call your peaks, because some peak callers ask if you want to look for broad peaks or narrow peaks. And so for Atec-seq, you kind of need both, which is why I actually use Homer to call Atec-seq peaks, because it is based on DNA-seq, which is kind of an old school way to look at open chromatin. And so a lot of those pipelines that were designed for DNA-seq are now being used for Atec-seq. This is very different from Chipseek peak calling, where you have an input sample that you also run in addition to your Chipseek. So when you shear your chromatin, before you do the immunoprecipitation, you're going to take a small amount of that and run it on the sequencer. And that's going to be indicative of just kind of all the fragments in the genome, right? And so when you then do your Chipseek, you want to look at the number of reads in a particular area that are enriched compared to your input sample. So this would be called a peak because there's way more reads in this area compared to input. As you can see here, I have another example that wouldn't be called a peak because for some reason, either the DNA, for a number of different reasons, you can have a buildup of sequences in your input that are not enriched in your Chipseek. So these would be, a lot of these are regions that are kind of blacklisted, and you would remove these from your analysis. They could also be as a result of PCR duplication, right? Which they should have been removed in your quality control step. So anyway, you have your peaks that are enriched in relationship to input. And again, these peaks can be broad if you're doing like a histone mark chip, you know, for H3K4 trimethyl, which is indicative of active transcription. These peaks would be generally large. However, again, if you're doing Chipseek with a transcription factor, these peaks would be much more narrow. And overall, you would see much fewer Chipseek peaks versus Atec-seq peaks. And this is just due to the nature of the experiment, right? In Atec-seq, you're looking for all the regions that are open, whereas in Chipseek, you're just looking for specific regions of DNA that are bound by one protein of interest, right? So theoretically, if you did enough Atec-seq, it would be like a Chipseek of every single protein that's bound to the genome, but that would be a like really high resolution that we can't obtain right now. But so you're going to get way more Atec-seq peaks than Chipseek peaks on average. So this is an example of what this data might look like in real life. So as Sarah pointed out in our introduction, our lab studies congenital heart disease. And so we use the IPS cell model and differentiate these cells into cardiomyocytes over a 30-day time period. And so when you do this with just wild type cells, I've performed an Atec-seq time course over cardiac differentiation. So we've got Atec here on the top, and we've got Chipseek for histone marks on the bottom. And as you can see, we're at a pretty big region of the genome, about 250 kb, and you can see these regions of DNA here that are open at various time points. And you can see over time that some peaks stay the same, and then some peaks appear and disappear based on time. These peaks, I think, look pretty nice. They actually should look like peaks and not boxes, like I kind of showed on the previous slide. And they should have a minimum number of reads. So if your peaks look like boxes, they're generally probably not real peaks. And the peak caller will take this into consideration because it's usually PCR duplicate or just a minimum too few reads to establish whether this was a true peak or not. I will point out also that as you differentiate cardiomyocytes over time, sometimes the cells kind of go and differentiate into, there are multiple cell types in a cardiac differentiation. And so therefore, like at day 4, we mostly just have mesoderm. And so these peaks are much cleaner. They're much higher than some of the peaks that we see here at day 30. You have higher background. And this is relatively normal in a more complex cell type. And actually now single nuke Atec-seq is available where you can kind of look at these peaks in different cell types at single cell resolution. But this is just bulk Atec-seq data. This is also, it's nice to compare regions of open chromatin by Atec-seq to regions of enhancer activity, for example, with H3K27 acetylation Chipseek. You can see that these peaks do overlap and that the Chipseek data is a little more broad than the Atec-seq data if you're looking at broad peaks like histone marks. All right. So now that we have our peaks, the next thing we want to do is assign them to a gene, right? Because we want to look at expression of these genes. And so we need to associate a peak with the gene. And I actually think this is the hardest part of the analysis, is to assign a peak to a gene. And there are a lot of annotation tools out there. A lot of them use the nearest binding or the nearest gene method. And so what this would look like, for example, here's a schematic that I took from the Harvard GitHub. We have our peaks here and we have our genome here. So we've got a blue gene, the promoter, and then the end of the gene. We've got a yellow gene here and a red gene here. And if you assign a peak to the nearest gene, this peak here would be assigned to blue, right? But this could be kind of misleading depending on the distance between this peak and this transcriptional start site. Because if this was a short region, maybe only 10 kb, for example, this peak could actually be a promoter peak for this gene. So some callers will annotate not just to nearest gene, which would assign this peak to blue, but it will look at the distance from the transcriptional start site of the nearest gene, and it will calculate or annotate based on that. So for example, if you use a different caller, this peak might actually be assigned to this gene here. So you just want to make sure when you do your annotation that you're consistent among all of your cell types. So I use the same annotations for Chipseek and Atec-seq. And you kind of want to just keep it in mind. Okay.
Lauren, there was a question
Sure.
about the input for the single cell method and how basically how that single cell method deals with uh, achieving the number of cells you need for good data.
So that's an excellent question. And unfortunately, I didn't talk about single cell Atec-seq data in this presentation. It's a relatively new technology, but basically what happens is just like single cell RNA-seq from our talk last week, single cell Atec-seq is at much lower resolution. And what happens is you cluster the cells by their nearest neighbor, exactly the way we learned last week for RNA-seq. And then those clusters are combined to call peaks. And so even though each region may only have 1 or 2 reads per cell or per nucleus, when you put them all together, you can then say, okay, well, there's 2 in every single cell in this cluster, and that is enriched over the regions around it. So it relies heavily on clustering and combining of clusters to call peaks.
Interesting. And then there was one other question just about your last slide, which is whether you find the TSS assignment better than the nearest gene assignment.
I wouldn't say that they're better. I would just say that they're different. So the package that I use here, I guess I'll talk about it a little bit on this slide. It really depends on the distance to the transcriptional start site, right? Like in the example I gave on the last slide, if it was only 5 kb from the transcriptional start site, I would assign that to the promoter probably. However, if it's like 100 kb away from the nearest transcriptional start site, and instead it's kind of in like a later intron of the gene upstream, I would probably assign it to that as well. So the program that I use here, and I'll talk about on this slide, is called Chipseek, which is an R package that will annotate these genes, and it's based on the nearest neighbor method, but you can provide parameters to specify a maximum distance from the transcriptional start site. So I would say, if it's more than 50 kb from the transcriptional start site, don't assign it to the promoter, assign it to the gene that it's in the intron for. I hope that helps. So that's what this package does, and it's one that I recommend using. And so it provides, Chipseek also provides really detailed information about the genomic region. So it'll give you the distance to the transcriptional start site, for example. And if you decide that, in an Atec-seq data set, if most of your peaks are not assigned to promoters or near a promoter, then your annotation is probably incorrect, or your experiment didn't work. In a typical Atec-seq, you would see, because the regions are the most open, most of your peaks, a majority of your peaks would lie within 10 or within 1 kb of the promoter. So this is an example of a pie chart where Chipseek has prioritized the annotations for each gene, and then you can plot as a pie chart the location of all of your peaks relative to gene bodies. So since some annotations overlap, Chipseek, for example, could assign it to promoter of this gene or intron of this gene. It will prioritize based on these rankings. So promoters always going to have first priority, and then the regulatory UTRs, and then the gene bodies, and then intergenic peaks, because there are a lot of peaks that are kind of really far away from the nearest promoter, and therefore it may not assign it to a gene. It might just say that this is a really an intergenic peak.
Lauren, there was another question from someone who's been looking at single cell Atec data, and they often see that the promoter of the nearest gene is actually itself not a peak. And so just wondering if you have any intuition for why the promoter of an expressed gene is more obviously open for bulk Atec.
That's an excellent question. I think it's just a numbers game, right? So if you're at a promoter of a gene that's turned on average, Pol2, Pol2 is not necessarily bound at every single promoter in every single cell in every single tissue type, right? So even if the gene is turned on in a tissue, it may not necessarily be turned on in every single cell. I think it's just a numbers game. And to be honest, like the single cell and single nuke Atec is a relatively new technology, and I don't have so much experience yet with analyzing that data, but I would be really interested to compare potentially single and bulk Atec data from a similar tissue type. That would be a really good experiment to do. Okay. So now we have our peaks, right? And we have our peak assignments, and we have our genes that are associated with that peak, right? So we're almost at the part where we're going to integrate our data together. But first, I want to talk about potentially overlapping different types of chromatin data together. So if we have Chipseek and Atec data, for example, how can we overlap these two? And since this is a primer, I figured I'd give kind of a little basic overview on how you would overlap peaks using a program called BedTools, which is available on the Broads cluster and HMS cluster. And everyone here, it would be available in your computing cluster. And it's a relatively simple command that I wish I had learned before I became a postdoc when I was doing some of these comparisons as a graduate student. So it's a very simple command. It's just BedTools intersect, and you provide your two peak files that you're interested in overlapping. So here would be your Atec file, and then here would be your Chipseek file, and then you can output this into an intersect file. And so this intersect file will contain all the regions in file A that overlap with file B. So as an example, this would be if you were to overlap these two bed files, this would not be an Atec-seq peak that overlaps with a Chipseek peak. So here in red are Atec data from wild-type cardiomyocytes, and here is a Chipseek for GATA6, which is a cardiac transcription factor. And so this would be an example where the Atec-seq and the Chipseek don't overlap. However, this would be an example where an Atec-seq and a Chipseek peak do overlap at a promoter. And we do see they don't overlap perfectly, right? But they do overlap. Here would be another example where these two data sets overlap. And here would be a third example where GATA6 is only bound in the Chipseek, but this is actually a region of relatively closed chromatin based on the lack of an Atec-seq peak.
Lauren, there is a question, sorry, just to go back to your last slide about what about enhancers? Why aren't they in your rankings of output from?
So we tend to prioritize, so let's go. So we tend to prioritize enhancers as intronic regions, and so they would be included here. So they are here. I guess I can make that a little more clear next time I give this talk. So thank you for input. But yeah, promoters would be the highest priority, and then within a gene body, introns would be prioritized as well. And actually, Chipseeker will break down which intron your peak is in. So for example, usually the first intron is where many enhancers are located. And so it will break down, you can isolate enhancer peaks by subsetting your data set to look for peaks that are just intronic or specifically in the first intron. It's kind of one of the reasons I like this annotation program too. It's really specific about where in the genome it's located, which exon or which intron or literally the distance in base pairs from the transcriptional start site. So that's why I use this program. Okay. So we have our peak lists, right? And we've overlapped our Chipseek and Atec-seq peaks to learn a whole bunch about open chromatin and where proteins are bound in the genome. And now what we want to do is look to see if any of these peaks are associated with differential gene expression, right? So it's kind of the lead up to the rest of the talk is how we can overlap Atec and Chipseek with RNA-seq. And again, I wish I had learned this as an early grad student, so I'm going to talk about it here in the primer, which is what this is for, and talk a little bit about the in command in R, which will take one gene list and return all of the genes that are in a second gene list. It's really straightforward. It's just one command. So here, say in R, you've annotated your peaks, you've done your DE-seq or your differential expression analysis, and you have two lists of genes. All you would do is ask which genes in your RNA gene list are in this Chipseek gene list here. And when you hit enter, it just gives you the list of all the overlapping genes, which is really nice. You can also look for genes that are unique by adding this exclamation point. So this would be the not command in R. So you are asking in this example of the genes in your RNA gene list, which of these are not in your Chipseek gene list? And so when you input that, you would get the genes that are unique to your RNA-seq data. And then if you reverse this, you would get the ones that are unique to your Chipseek gene list. And I will point out that in like most programming, capitalization is really key. So if you were comparing, say, human genes and mouse genes, or if you use different annotators, which I don't recommend, but TBX5 that's all in capitals would not be equal to TBX5 with just this, the first letter capitalized, which is also not equal to maybe a TBX5 without any capitals. So you just want to make sure that all of your gene lists have the same capitalization pattern before you do your gene overlaps. All right. So I spent the first little bit of time talking about Chipseek and Atec and how we can integrate this with RNA-seq analysis. I'm now going to go back and talk about Hi-C, which is pretty different than Chip or Atec, so I didn't talk about them at the same time. So we're going to talk now about Chromatin Capture and how we can use Hi-C data to kind of bring all of this data together. So there are a lot of methods of chromatin capture. There's 3C, there's 4C, there's 5C, now there's 6C, and there's also Hi-C, which is the one I'm most familiar with, so it's what I'm going to be talking about today. So so Hi-C is actually probably closest to Chipseek in the way that you cross-link your chromatin. So basically what chromosome conformational capture is doing is in the cell, we have, say a promoter that's brought to an enhancer, and those regions of DNA are physically touching to a certain extent, right? And so when you cross-link your cells, you want to cross-link the regions of DNA that are physically touching. And so when you cross-link your cells to get regions that are physically touching, and then you would do a restriction enzyme digest and add a biotin mark, a biotin tag to your DNA of interest. And so basically what happens here, you have your two regions that are touching that are separated by your proteins, right? So these are cross-linked together, and then you have your two DNA fragments. So you do your restriction enzyme digest and add your biotin to these ends, right? And then what you then do is a ligation step that will ligate these biotinylated ends together, or these DNA ends together. So now you don't need the proteins that are physically keeping them together. You've actually physically ligated your DNA together. Then you remove your proteins by reversing the crosslink, and then you shear by physical sonication, similar to Chipseek. So now you have your DNA fragments that look like this, and the ones that are physically next to each other in two-dimensional or in three-dimensional space, but not necessarily next to each other in two-dimensional space, are going to be biotinylated, right? So you've got your sequences here. These fragments can then be isolated using a streptavidin encoded bead. So streptavidin recognizes the biotin tag and will pull it down. And then you can generate your Hi-C library in a very similar way. So you add your adapters, you do sequencing of these, next generation sequencing of these fragments. And I will say, here's another big difference between Hi-C and all of the other techniques. So here, even though you only need about 10 million cells to do this experiment, you need to sequence at a really high depth. And this is because there are a ton of DNA-DNA interactions. And so if you don't sequence deeply enough, you're not going to get the statistical power in order to call a junction or not, or a peak or not. And so you need at least like 1 billion reads per sample. So this is not a trivial experiment in terms of time or cost, but you can learn a lot of information from this data.
Lauren, there was a question just going back to your previous overlap. Someone was wondering what kind of proportion of RNA versus chip versus Atec peaks do you generally see in cardiac tissues?
So like the percent overlap?
Yeah.
So generally, it really depends on your Chipseek antibody, right? And because it depends on the biology, right? So I'm going to give an example later of where the Chipseek and the Atec-seq actually don't overlap very well, but they do. The RNA-seq data kind of explained why biologically the Chipseek and the Atec-seq data don't overlap. So like I said, it really depends on the question you're trying to answer, but generally, if you were to do like a Chipseek with a H3K27 acetyl, like an active enhancer Chipseek, and then look at the Atec-seq, you get a really high, maybe up to 90% overlap. And that kind of shows that the quality of your data is good too, right? Because you're using two different techniques to kind of answer a similar question, like which regions are active. So the Chipseek, I guess, is the wild card there. But generally, if you have like differential Atec-seq, a lot of times that will be associated at the promoter. So if you have differential Atec-seq peak at the promoter, a lot of times, I would say maybe 80% of the time that it's associated with differential gene expression in those cells. Okay, so yeah, I guess I just want to reemphasize that the chromosome conformational capture, high C, doesn't require more cells, but it requires really deep sequencing to generate the maps that you need to perform these analyses. One last thing I want to say is similar to Chipseek, you can do this on frozen tissue, but the cells must be cross-linked before you freeze them. Otherwise, it won't work. It is less likely to work, we'll say that. And then the sequencing resolution, again, is going to be, it's also different from Chipseek or Atec-seq. This is at low resolution because these DNA contacts are very, I would say temporary, I guess. And so what happens, you need a lot of sequences to build your DNA junctions. And so you can really only do that at relatively low resolution. And I'll explain that a little bit more on the next slide. So again, this is just a different schematic now coming from your cross-linked chromatin, and now you have your fragments, and then you've sequenced them at really high depth, right, to billion reads. And so the next step is to align them to the reference genome. And this is more difficult than Chip or Atec-seq because you have sequences of DNA that aren't, they're connected in a three-dimensional space, but they're not necessarily linear DNA, right? So you want to make sure you use an aligner that's really good at with gap sequences. And so I recommend BWA-MEM for this. And so when you align your sequences to a reference genome, the next thing you want to do, so high C is paired end. And so the first end is going to be the one end of the junction and the second end is going to be the second end of the junction, right? So they're going to map to different regions of the genome. And you're going to have to put this back together in order to get the interaction, where the DNA pieces are connected, right? So you want to pair back your forward reads and your reverse reads to form these paired read files that you can then use to bind together to generate a high C map, which I'm going to show you on the next slide. So again, the resolution of your experiment here, it depends on how big your bins are going to be. And the literature really recommends 5 kb to 10 kb regions. And actually, I forgot to mention, I'm sorry, when you want to pair your reads back to each other, there are a lot of now downstream pipelines that will help with this. So in addition to using an aligner that's pretty good at gap sequences, there are a ton of tools out there that will help you kind of reassemble these reads. And I recommend the Juicebox toolkit. The program that you use to look at chimeric reads would be called Juicer. And they have a web browser version, which is kind of nice for people who aren't so informatically inclined, but they also have a package that you can just download onto your laptop. So I find it really, it's a really great tool and I highly recommend it. So now that we've paired our ends and filtered and binned, we can generate a high C heat map that looks kind of like this. And so again, the goal of high C is to look at long range and short range chromatin interactions. And you can do this by analysis of this data. You can get what's called loops and what are called TADs. So these are kind of, this is kind of a hot topic right now. So TADs are topologically associated domains. So they are large regions of the genome that interact with each other, but not other TADs. So for example, genes within this TAD here are significantly less likely to interact with genes or with DNA sequences within this TAD. So these are large regions of compartments that interact only with each other. And you can learn this from the high C data, right? So TADs are demarcated usually actually by the presence of CTCF, which is a protein transcription factor, I guess. So CTCF Chipseek would be really helpful here to establish and validate where your TAD boundaries would be. So this would be a nice way to incorporate high C with Chipseek data to kind of get a better understanding of where your TAD boundaries would be. And then within the TADs, you have what are called loops. And so these loops are regions where pieces of DNA interact with each other inside the TAD, right? So the way high C data is kind of presented, so you have your genome actually on the X and then your genome, the same genome on the right, and you actually plot which regions on the X-axis will interact with regions on the Y-axis. So, and they will be assigned a color based on the bin, right? So if you have more smaller bins, there are going to be more squares here with different colors. But if you have larger bins, your colors are going to be, or your square sizes are going to be larger, right? So, right. The main point of this is to show, okay, you have your TADs, where regions of chromatin interact with each other within the TADs, within loops. So these little tiny squares would be regions where DNA interacts with one another, right? And so it would be really helpful to have the Chipseek data to overlay with the Hi-C to show the locations of the TAD boundaries, and that genes that are within one TAD are more likely to interact with each other, but not with regions outside of the TAD. And so if you want to look at RNA-seq data, for example, it might be beneficial to look at regions within these TADs and not just one gene, because maybe all of these genes are misexpressed because there's a problem with chromatin looping, for example. So it would just be a good thing to keep in mind that like you might want to look at multiple genes within these loops, not just one. All right, so in the last few minutes, I want to give two examples of how in the real world and we can utilize all of this data together to tell a really comprehensive story. And so I have an example here of a paper that we just published in eLife last week, where again, our lab works on congenital heart disease and cardiac development. And so we found, we were working with this gene called GATA6, which is a known congenital heart disease gene. And so I had done a Chipseek for GATA6 and found that we got about 20,000 peaks in our genome, which is pretty standard for a transcription factor. And then when I performed Atec-seq and overlapped the two gene sets together, I was at first kind of surprised to discover that the majority of GATA6 Chipseek peaks do not overlap with open chromatin. So here's kind of a schematic of what this would look like. We have our open chromatin. Only about 12% of our genes our peaks, overlap with an Atec peak. The overwhelming majority of peaks of GATA6 and Chipseek do not overlap with Atec-seq data. And the next thing I wanted to ask was to be like, okay, GATA6 is bound to these regions. Does it affect expression of the genes within these regions? And so what I had done was looked at the differential gene expression and found that of the 3,000 genes that were differentially expressed between two time points, we get about 600. So a pretty high percentage of the genes that were bound by GATA6 at closed chromatin had differential gene expression over time. And so actually the conclusion that we drew from this, and we did some other studies to show, was that GATA6 is actually a pioneer factor in cardiac development. And so GATA6 will bind to closed chromatin and find a way to open the chromatin, so it either recruits a helicase or something like that. We actually don't know the mechanism by which we do this, but we think it's through recruiting other factors. So GATA6 binds to closed chromatin at day 4 of differentiation, and then by day 5 of differentiation, those regions are now open and are differentially expressed between GATA6 wild-type cells and GATA6 mutant cells. So it was only after incorporation of the chip and the Atec and the RNA-seq data that we were able to determine that GATA6 was indeed a pioneer factor for development. So this paper, again, that was kind of the main conclusion of this paper, actually, that was that GATA6 was a cardiac pioneer factor, and that mutations that we see in congenital heart disease patients actually affect GATA6's ability to actually be a pioneer factor. So that was a really exciting study that just came out last week, so I encourage you to check it out. So I guess to wrap up real quick, each data set on its own can provide one piece to the puzzle. So we can look at open chromatin by Atec-seq, transcription factor histone binding by Chipseek, long and short range chromatin interactions by Hi-C, and then differential gene expression by RNA-seq. And it's really the integration of all of this data together. And now that next generation sequencing is relatively accessible for everyone, integration of these data sets together can provide a greater understanding of gene regulation. And so that was all I had. I'd like to acknowledge my advisors, John and Cricket, and everybody who works in the Simon lab. I was a developmental biologist before I started getting into the chromatin biology, and so I learned a lot about informatics from them. And I'd like to thank our collaborators as part of the Pediatric Cardiac Consortium. They give us the interesting genes to study. So I'd like to thank everyone, and thank you for your time.
Thank you so much, Lauren. I think we probably have time for one question, which just came through. And the question was how to get differentially expressed accessible regions with Atec-seq.
That's a great question. So what you would want to do is, so say you have a wild type and a mutant, right? So let me try and go back to some slides here. Instead of this being Chipseek, let's pretend that this is Atec-seq. And so you have Atec-seq for a wild type and you have Atec-seq potentially for a mutant. So you can do differential peak expression analysis for both Atec or Chip by comparing a wild type and a mutant, or 2 time points, for example. The peak calling programs, Max2 or Homer, have parameters where you can compare 2 samples to each other. So you would compare this sample to this sample, and you can provide it, say, I want to know every region where this peak is 4-fold higher than this peak, for example. So this is all done in the peak calling software. And like I said, you can use either Max2 or Homer to do this. And I find them to be pretty good. It depends on how good your Chipseek or your Atec-seq is. And you want to make sure that your parameters, you test a couple different parameters. Some people use 4-fold, some people use 2-fold, some people use just Q of an adjusted p-value of 0.05. So the literature is kind of still out there. The debate is still out there about which is the best parameters to use, but I think they're very dependent on your data set.